Hello everyone, uh, I'm Ashfaq Ahmed and I'm PhD in Bioinformatics and Structural Biology. Before today's video, we have covered part 1 of phylogenetic analysis where we have discussed introduction of phylogenetic tree. There were some names and some annotations, how to read, how to look like, how you will read, how you will analyze, and how you will understand the phylogenetic tree. So today is part 2, and in this video I will talk about phylogenetic inference, or we will also call it phylogenetic algorithms. Why do you need to understand these methods? Most of you may be confused, like why are we are going to understand about these algorithms, because you will not perform this. Actually, the computer will perform this, but you need to understand this. And if you understand these algorithms briefly, you will be able to decide which method to select for what kind of data. So let's move. Broadly, there are two approaches in deriving a phylogenetic tree. When you are constructing a phylogenetic tree, there are many approaches, but the famous one, there are actually two famous approaches. The one is called phenetic approach, or we also call it distance-based approach. In distance-based approach, actually the computer or the algorithm will look for the distances between two or more sequences, and based on those distances they will cluster the sequences into a separate nodes, and ultimately they will make a tree for you. So likewise, I have mentioned here, like, proceed by measuring distance between sequences and generating the tree by hierarchical clustering procedures. It's very simple, just like and these methods, the distance-based methods, they do not calculate any historical models or any historical relationship. It doesn't matter if the sequence is from which background or from which species. If you select a distance-based method, the computer will only calculate the distances between the sequences. Now in next slide I will tell you what the distances actually means. So consider you have 4 sequences in your data: sequence 1, sequence 2, sequence 3, sequence 4. Consider you want to generate a phylogenetic tree, and you have selected a distance-based method or phenetic method. So the first sequence is ATCC, the second sequence is ATGC, the third sequence is TTCG, and the fourth is TCGG. Now the computer will generate a kind of matrix during the construction of phylogenetic tree. And how this matrix will work, you need to understand this. The 4 sequences I write it here, like sequence 1, sequence 2, sequence 3, sequence 4 on the top panel, and also here sequence 1, sequence 2, sequence 3, sequence 4 in the same order. Now look for distances, like ATCC. When compared to its own ATCC, there is no difference, so the distance is 0. Now when this sequence ATCC is compared to this one's ATGC, so there is one distance. How? If you compare these two, A with A, T with T, C with G on the third position, so they are apart with one mutation, G and C, and the fourth one is again C and C. So both these sequences, this is actually sequence 2, this is actually sequence 1. So both sequence 2 and sequence 1 are far away from each other with distance 1. It means the more mutations, the higher the distance. Okay, and if we compare sequence 1 with sequence 3, it has two distances, distance value of 2, and if we compare sequence 1 with sequence 4, it has distance value of 4. So based on your data, like in this example we have totally 4 sequences. So based on this data, the smaller distance, the sequences with smaller distance with each other will be clustered together. So after analyzing this kind of matrix, you can see, so we came to a conclusion that sequence 1 and sequence 2 contain the smaller distance 1. You can see, because here is 2, this is 3, this is 2, this is 3, this is 4. So the smallest distance is between sequence 1 and sequence 2. So what will happen? In this case, the computer has decided that sequence 1 and sequence 2 are very similar with each other based on the distance calculation. So the ATCC sequence 1 and ATGC sequence 2, they both will group on the same node. Now how they will calculate the fate of sequence 3 and sequence 4? The same matrix will be reduced and we will call it reduced matrix. Once again the computer will calculate it, you will not calculate it. You're calculating this matrix, but you should know how the computer will treat your data. So now for sequence 1 and sequence 2, we are now clear that they will take a similar node because they have lower distances with each other. So let's see. So now this case has been solved, so we treat it as a single sequence, or the computer treat it as a single sequence. Now again a reduced matrix. Now the sample became 3, 1, 2, 3, but in sample 1, in sequence 1, you can see this is sequence 1, sequence 2. It means both these sequences are taking the same node. So write it like this is the combined node, or this is the sequence 3, or this is the sequence 4, and the same pattern you can also draw it by yourself. Now let's analyze it. This is the new sequence 1 or against its own, so you can see a 0 distance. Okay, this is now how you will compare, or how the computer is comparing this node or this sample with this one, because here is only one sequence 3 actually, and here is sequence 1 and sequence 2. So look here, ATCC, ATGC, actually I write it the same thing here, and during comparison this sequence will be compared to both of this and this. So now see, TTCG is this sequence, and it will be also compared with this TTCG. So if you see, here is a mutation and here is a mutation. So this is 2, this distance will becomes 2 plus 1, 2, 3. So this part has 3 mutations and this part has 2 mutations. So 2 plus 3 divided by 2, because we will take an average. So if this is, there are 2 sequences, like this is sequence 1 and this is sequence 2, so the average is 2.5. The same way you can calculate for this, and the distance will be 3.5. Now when sequence 3 was analyzed, so the distance came 2. So in the reduced matrix, if we see the sequence 3 comes with a smaller distance, that is 2. The other distances are greater, like 3.5 and 2.5. So the sequence 3 will take a node with sequence 4. Okay, now the case has been solved and we will have this kind of tree: sequence 1, sequence 2, sequence 3 and sequence 4. The sequence 2 and sequence 1 will share a same node because both of them are showing they have shown smaller distance, and the sequence 3 and sequence 4, both of them will group to a individual node. But how these branch lengths are calculated? The formula is very simple, and let me teach you. The branch length of h between node x and y will be calculated as 1 by 2 distance between x and y. It means distance between this node and distance between this node will be summed up or then divided by 2. Remember, if we have 3 sequences here, so the distance between them divided by 1 by 3. Okay, so go back. What was the distance between sequence 1 and sequence 2? It was 1. So 1 and 1 by 2, so 0.5. This will be the branch length. Now sequence 3 and sequence 4, what was the distance? 2 divided by 2, it will be 1. Okay, now this distance, how this distance comes up, like 1.5 and 1.5, that is very simple. Sum up all these distances and divided by 2, because we have 2 nodes. So this is 1, this is 1, this is 0.5 and this is 0.5. So sum up 1, 2 and 3. We have 2 nodes, 1 and 2. So divided by 2. The mean will be 1.5 for this one and 1.5 for this one. Okay, I hope you got how the distance methods will treat your data and how they will calculate a tree for you. Remember, there are 2 famous methods. One of them is called UPGMA, that stands for unweighted pair group method with arithmetic mean. Also neighbour joining. Both of these methods are actually distance based methods. Remember, the distance based methods, they do not account for any evolutionary history. They do not account for any real evolutionary history. They only take care of distances. So we will talk in the final slide that what kind of matrix you have to select for what kind of data. So I hope from now on you will be able to understand, if you are selecting UPGMA method or if you are selecting neighbour joining method, you will know that these are the distance based methods. So second approach is cladistic approach, and it considers possible pathway of evolution. They do not consider only the distances, they also consider the evolutionary pathway. How? I will teach you. Okay, it infers the ancestral feature, it cares for the ancestral features, but the previous one we did not talk about any ancestral feature, we just talk about distances. Okay, and it chooses an optimal tree according to some model of evolutionary change. It means when you are dealing with cladistic method, the program will design or will construct many trees, and then it will give you the optimal, the best one. Okay, there are 2 methods you need to remember these names. names maximum parsimony and maximum likelihood. So whenever you are constructing a tree, and if you have selected maximum likelihood or maximum parsimony, you should know that you are working with cladistic approach, and this approach consider the evolutionary possibilities or the evolutionary pathways. But if you have selected the other two, neighbour joining or UPGMA, so remember they will only construct a tree based on the distances. Okay, so how this method works? Suppose once again we have 4 sequences: sequence 1, sequence 2, sequence 3 and sequence 4. So the program will try different combinations. For example, in this case the program will try 3 combinations, but on the slide you can see 2 combinations. That's okay. I actually want you to give you a basic idea. You will understand even from these 2 combinations. So how? The program will first construct a tree without knowing any distance. It will keep sequence 1 and sequence 2 on the same node. This is the node, and this is sequence 1, this is sequence 2, and it will keep sequence 3 and sequence 4 on another node, and then the program will calculate the mutations. For example, in these 2 there is only one mutation, that is C at position number 3 to G. You can see it here, C to G, and the program has constructed a hypothetical ancestor ATCG of these both sequences ATGG. So the same way the program has calculated ancestor, and there is only one mutation T and C, and then they have constructed the real ancestor, I mean the final ancestor. You can see ATCG and TTCA, and that came ATCA. Okay, so now count the mutation: 1, 2, 3, 4. 1, 2, 3, 4. So solving this tree, the program counted 4 mutations. Now the program will shuffle the sequences. Sequence 1 will be grouped with sequence 3, sequence 2 will be grouped with sequence 4, and now the same thing will be repeated. So in this combination the program observed 1, 2, 3, 4, 5, 6, 7 and 8. The program has detected 8 mutations. Now in the third, the program will try sequence 1 with sequence 4, sequence 2 with sequence 3, and once again they will count the number of mutations. So remember, the maximum likelihood or maximum parsimony, they will prefer a tree with the least number of mutations. So if we talk about these 2 mutations, these 2 trees, so this one is the optimal tree, not this one, because this tree supports 8 mutations and this tree supports 4 mutation, 1, 2, 3, 4. So these both these maximum likelihood and maximum parsimony, they will you will understand now. You got my point, that the maximum likelihood and maximum parsimony methods actually draw many trees and then search for an optimal tree which supports the lowest number of mutation. Also, this method constructs an evolutionary ancestor which was not present in the previous one. So the previous one, the distance-based approaches only consider the distances, doesn't matter what kind of ancestor they have. But the second one has 2 differences. Number 1, it creates a hypothetical ancestor sequence. Number 2, it generates many trees and then decides the optimal one. So you should know from these two conclusions, okay, what you should know. Maximum parsimony and maximum likelihood holds more accuracy than the neighbour-Janning and UPGMA. So remember, if you are dealing with your data to construct a tree, so if you need more accuracy, then never try UPGMA or neighbour-Janning. Try maximum parsimony and maximum likelihood. But remember, the maximum parsimony and maximum likelihood will be slower compared to neighbour-Janning and UPGMA. So if you want your data to be treated faster, then I think neighbour-Janning and UPGMA is the better option. But if you want more accuracy, then it will be slower, because I told you the maximum parsimony and likelihood will generate many trees and will give you the optimal tree. Okay, how will you decide that which method I should choose? Now you have to look your data. It's up to your data. For example, if you have 10 sequence, 20 sequence, 40, 50, that's okay. I think you should go for maximum parsimony or maximum likelihood, because nowadays the computer power is good enough with everyone, so it will not take so much longer. So the number of sequence, that from the data I mean the number of sequences you have in your data. If you have many sequences in thousands, then you should think about it. Or the second variable is the size of sequence. For example, if you are dealing with coronavirus data and the whole genome data, so as for my information, the corona genome is composed of almost 29,000 nucleotides. So remember, if you have 29,000 position in one genome, and you have, let's suppose, 1,000 samples, if you have 1,000 or more sequences, so it will took a longer time. So in that case, first I would suggest to try UPGMA and neighbour-Janning, because that will be quicker as compared to the other. But remember, the neighbour-Janning and UPGMA do not consider a real evolutionary stuff. They only give you the evolution based on the distances, or you would say based on the mutations. So at times, if you are dealing with DNA data, so there are also codon degeneracy, and we call them in protein, we call them synonymous mutations. So the distance based methods like neighbour-Janning and UPGMA, they will create a distance, but in reality it will not be a distance. Anyway, okay, the second part is hypothesis. Your supervisor must have guided you for certain stuff, like to do this. So actually from the hypothesis, I mean that actually what you want from your data. So based on your hypothesis, you have to decide which method I have to consider. And the final one is the computational power. What kind of system do you have? How long you can keep it in the start mode, and what kind of charging you have. So all these things considered, and specifically for those who has electricity problem in some areas they have. So it was all about the inferences and algorithms. Stay tuned with me, and in next video I will practically show you how to build a phylogenetic tree, and then how to present it for your thesis and for your reports. So for now, thank you. Thanks for having me, and if you have any question, please drop in the comment section, and subscribe this channel and share it with other members. Thank you. Bye bye.